Hi everybody, Mr. Trachtenberg here with a little walkthrough of the Strawberry DNA Extraction Lab that you can definitely try at home. Safety first, of course. Not that anything here is super dangerous, but almost anything, if it gets in your eye, can at the very least be a nuisance, and at the worst cause you a lot of pain. So I wear regular glasses, safety goggles, just in case. I'm not really going to worry about gloves because we don't have anything that caustic that's going to cause issues. So, what do you need? You have the lab. One, I always like to print mine out. It's a lot easier and less stressful than having your laptop right there. You don't have to worry about getting liquid or strawberry mush all over it. And yeah, so if you happen to have a printer at home, I highly recommend printing it out. If not, it's not that long a lab. You could easily write down the steps and have them out in front of you. But, so what do you need? You need a heavy-duty Ziploc bag, just like this is fine. Strawberry, fresh or frozen. This happens to be fresh. Coffee filter, like this. If you don't have any coffee filter, you can take some paper towel and use that. It pretty much does the same thing. A plate. And really, if the plate is just to cut down on the mess so that you're not necessarily cleaning up the counter. Test tube or clear glass cup, baby jars or baby food jars. Really anything that you have that's see-through can work. You don't want like a low flat dish though. Something that's a little bit more taller than it is wide would probably work better. And a glass rod, pipette, or straw. This is like a little kebab skewer that I'm going to use, but you could easily use a plastic straw or a metal straw, even a toothpick for that. Okay, so in your reagents or chemical list, you've got the list of ingredients you need for making your DNA extraction powder. I took the liberty of doing that before the video just to cut down on the length, but basically it's about 1 teaspoon, and it doesn't have to be exact for this, of that's about this much, dishwashing detergent. It says ivory, but that's just because ivory is clear. So if you have clear dishwashing detergent, that's the best, just because it won't interfere with the color of the strawberry, but that doesn't really matter. I used palm olive, which is green, so I get a slight tint there. Table salt. I like this because it doesn't have iodine added in. It's just one less chemical. But any kind of salt, table salt you have is fine. You need a real small amount. Like this is a ½ teaspoon. You don't even have to fill that all the way up. Let's see, water, about 3 ounces. So this is a 4-ounce jar. So about 3 ounces of water. And mix it all up. And then you have your extraction buffer. So it's really just soapy water with a little bit of salt. And we'll go over what these things are needed for. So, and then ice cold ethanol or isopropyl alcohol, also known as rubbing alcohol, which you can get usually at a grocery store, dollar store, or any sort of pharmacy or drugstore. Or at least you used to. For a while, they kind of cleared off the shelves when quarantine and pandemic stuff first hit, but it should be coming back now. So, step 1 is to put your fruit in the bag and try to squeeze out all of the air because you don't want the bag accidentally popping open. And then you're going to smoosh up your strawberry until it's like a paste. I'm also going to do this concurrently with a banana because I have heard that the results are similarly interesting with a banana. So we'll give it a shot. So here's how I like to do it. Just kind of push down, and once you make, strawberries are pretty mushy to begin with, so once you get this far, it's pretty easy. You just kind of want to get rid of the big chunks. And this banana was super ripe, so I'm guessing it's not going to take. Oh yeah, that's already like a paste. Yeah. That's got banana bread written all over it. Well, not after what we're about to do to it. Okay, then, yeah, it says smush it for about 1 to 2 minutes. You don't have to time yourself, that's just to make sure that you've got as many small pieces as possible. So think about it though. Strawberry is a fruit, which means it came from a plant. And we want to get the DNA out of this plant, which implies that DNA is somewhere inside. So, how do you get the DNA out? Well, there's no button or snap or zipper or anything like that. You have to break it out. And plants have these things called cell walls around all of their cells. They give the plants their structure. In trees, they give them wood and trunks and stems. And smushing the strawberry like this breaks down those cell walls so we can begin to get at the DNA. Even bananas have cell walls because after all, they're part of a plant. Okay, now we're going to add 10 milliliters of extraction buffer. So this is it's like a little measuring cup that I have that has milliliters. So I'm going to hold this at eye level. Well, technically you're supposed to get down to its level. And then slowly pour in. About 10 milliliters right there. So I'm going to add... Not a big fan of the slider bags. Just hard to tell if they're open or not. And sometimes they don't open. Like this one. This could be bad. Alright, don't fail me now, slider. Alright, I'm just going to pull it apart. Technical bag difficulties. Alright, well, let's do the banana first. Okay, that one worked. Right? No? Okay. Note to self. These are not the best bags for this experiment. Come on. There we go. Okay, let's add that in. And hopefully it'll allow me to close it up again, because this is the real messy part. Okay, that looks pretty good. Now let's see if I can handle opening up the strawberry bag. I swear I've done this before. There we go. Okay, so let me get 10 more milliliters of this stuff. The extraction buffer. That's good enough. Doesn't smell great. Technically I didn't smell that properly either, but I knew what I was smelling. Okay, good. Okay, so now we do that, and now we mix it up again. And this is where you really want to get like a uniform kind of paste. It should all sort of look the same color. So the question is, you know, this is soapy water, so you're going to see some bubbles start to form. So what's the point of the soap? Well, soap breaks down fats and grease, which is why we use it on our used up dishes and pots and pans and things like that to get the grease off. But why would we need it then in a plant? How is this plant fatty or greasy? Well, the answer is it's not necessarily like the pulp of the strawberry, but all cells, even plant cells, have a cell membrane. Animal cells have a cell membrane too. What animal cells lack that plant cells have is the cell wall. So right inside the cell wall of the plant cell is a cell membrane, and that happens to be made out of fats or lipids. So what we're doing is we're using this soap to further break down the cell, strip away the outer membrane protection as well as the inner membrane that's around the nucleus that contains all the DNA. So the better you do this step, the better your results will be. So you do that. Now, here's where it gets messy. You take your collection jar and you're gonna pour out this mixture into your coffee filter and then squeeze it into the jar. And we're on to the second page of our lab. So you start like this, basically. Some people like to cut like a little hole in the corner of the bag and squeeze it out like frosting, and you can totally do that. It definitely works. Now, you don't necessarily need to get all of it out. What I have here is probably plenty. And then what you want to do is just kind of gently squeeze it through. You don't want to squeeze so hard that you break the filter open, because then the filter's not really filtering anymore. You're just getting big chunks of pulp in. And the whole point of the filter is to keep the solids of the strawberry out while the liquid parts and the juices of the strawberry, as well as hopefully the DNA, go in. So, Okay, and I never mentioned why we added the salt. What the salt does is when the DNA and other proteins of the cell are all mushed together, the salt helps strip away some of these other proteins that are attached to DNA that we don't want, and it kind of keeps them from coming out of solution when we're trying to just get the DNA out. So it sort of binds to them and keeps them happy. So here's really all you need. You don't need a ton of it, and it it smells nice, it looks nice, but it's not Jamba Juice, right? I mean, this is strawberry parts, but it's also a bunch of dish soap and salt. So it's not for drinking, it's not for tasting. If you want to eat a strawberry, you do that after the lab, after you're all cleaned up. This will, at the very least, give you some quality diarrhea, and at the worst could lead to vomiting. So save the experimentation for the jar and not the mouth. Okay, so now we'll go over the sink real quick. Rinse off